Greetings. I am Professor Pranab Goswami from Department of Bioscience and Bioengineering, IIT Guwahati. I am thankful to Northeastern Cell, NRDC Govtop India, IIT Guwahati, for giving me opportunity to deliver this talk. The topic is scope for innovation in the field of biosensors, with special emphasis on the state of Assam. So before discussing the topic, I would like to briefly describe what is biosensor. Biosensor is an analytical tool. It is a small, so that's why it is portable. It is simple. It's capable of using by semi-skilled operators or by patients themselves if it is for clinical applications. And preferably, it should be inexpensive. I think most of us are aware the biosensors which are available in the market, the glucose sensor and pregnancy test. So these biosensors are basically man-made device, but these are inspired by the natural sensors. So you might have seen these plants. When we touch these plants, the leaf of the plants are collapsed. This is one of the good example of pressure sensor. We did not have to go to the plant kingdom. Sensors are with us. We have Pancha Indriya. We have Shwadeno, we can taste. Graneno, we can smell. Dristi, Shravana, we can hear. Sparseno, the touch. So these natural sensors are omnipresent. It is not only in human being or in the plant kingdom. Even in the current situation, as you know well, the coronavirus spike protein does not recognize the cells. If the cell surface does not have the receptor, which is called AEC2. So if it does not get the specific receptor, the coronavirus cannot dock on the surface of our cell and cannot deliver its genetic material inside the cells for its reproduction. So these natural sensors are amazing. The major amazing thing in natural sensors is their biorecognition system, the way they recognize the system. So this biorecognition system of the natural sensors are highly selective that inspired the scientists to develop biosensors. So there are certain performance factors for the man-made biosensors. Those performance factors are one of the selectivity, that selective natures it acquires from the biorecognition element which it is used here from the natural sources. It gives fast response. Generally, the biosensor gives response in seconds. It is highly sensitive. It gives in sub-millimolar range. Even it can go to the femtomolar level also, depending on the demand of the target. It is accurate. It is reproducible. It is independent of physical parameters. So for operating the biosensors, we need not have to use pH meter, balance, and all this kind of laboratory equipment which generally we use. So here, the biosensors works independently without those equipments. And it has short recovery time. Basically, once the biosensor is used, the sensor surface should get ready for the next sample in a short span of time. And it should be functionally stable. Functionally means it's both operationally as well as shelf life of the biosensors should be high, should be prolonged. If you see all these performance factors, we can say that it is a decentralized testing facility. If it is for clinical application, then it can be used for point-of-care applications, testing centers, in home diagnosis. And if it is non-clinical applications, then it can be used for anywhere in the on-site applications. So it is suitable for remote locations in resource limiting environment. Now, let us come to the principle of detection by biosensors. As far as the biosensor configuration is concerned, it has few components. Among these, three very important components are: one is the bioreceptor, and other is the transducers, and the third one is the signal processors. So the bioreceptor is the one of the important component where this biorecognition element like enzyme, antibody, DNA, RNA, even cell membranes, or even antigen are generally immobilized over here. And these bioreceptors are always keep in close contact with the transducer. So the function of the transducers is to transduce the biochemical signal, which is generated upon absorption of the analyte target of your interest to the bioreceptor. And this transduce this biochemical signal to some other form of signal. This signal may be optical, electrochemical, piezoelectric, calorimetric, and this signal are ultimately again transduced to electrical signal, which is then processed, and finally it appears in a digital form to understand the quantity of the analyte which is present in a sample. So based on the type of transducers, the biosensors are generally classified. There are basically four different kinds of transducers are there. One is optical, another is electrochemical. Again, within electrochemical, different types are there, like amperometric, voltammetric, potentiometric, conductometric, and so on. Then piezoelectric transducers, which is basically involve change of mass. Then calorimetric transducers, which generally involve the change of heat or enthalpy change. Now these biosensors can be constructed. The question is that how these biosensors are fabricated or constructed. Certain materials are required for its construction. One of the important material classes, the nanomaterial is used, modern biosensor technology. Then various kind of polymers, conductive polymers, non-conductive polymers are used. Metals are used, highly conductive metals, if it is used for electrochemical biosensors, because electrochemical biosensors involves electrode. So these electrode materials, in many cases, highly conductive metals are used. Of course, nowadays instead of metal, highly conductive nanomaterials are also used. Then biocompatible support is required for its construction, because here we use the biorecognition element, which are enzymes or antibody, those kind of proteinous biorecognition elements are also used. And those biorecognition elements are usually very unstable. So to stabilize these biorecognition elements on the surface of the transducers, many biocompatible support systems are used. Various kind of redox compounds are used, and various smart materials are used. So one of the emerging materials which are commonly used in biosensor development is the graphene or its graphene-based or its derivatives. Graphenes are honeycomb structures with sp2 hybridizations. There are graphene oxides are also used. These graphene oxides are very useful for coupling the biorecognition elements so that can be immobilized on the surface of the transducers. The most important property which is exploited for developing biosensors from the nanomaterial is that their surface area is drastically increases from the bulk material. And so these materials, when converted to some kind of nanoforest, the immobilization of the biorecognition element is drastically increased. Basically, it increases the loading of the biorecognition element on the surface of the transducers. So when there will be more loading, more dynamic range will be there, and sensitivity of the sensor will increase. So here is the beauty of the nanomaterial, which is used on the surface of the transducers. Now, there are certain criteria that support the commercial success of biosensors. One is that multiplex detection facility. If the biosensors can simultaneously detect different analytes from a single species, for point-of-care application, this is very suitable. Another important criteria is that non-invasive clinical samples, such as saliva, sweat, tears, stool, urine, and all those kind of extra body fluid materials, because drawing blood from a human is a bit painful and patients generally do not like it. So if biosensors can be used for those non-invasive clinical samples, then that will increase its chance of to penetrate the market. Integration of wireless technology, such as mobile phone, Wi-Fi, Bluetooth, etc., because this integration helps to transfer this biosensor testing data to remote to centralized laboratory or wherever the user wants to transmit this data to get the feedback. For example, if it is for clinical applications, then data can be transmitted to a centralized laboratory to a doctor who can then, by looking the data, suggest the therapeutic intervention. So this modern integration wireless technology is very important for disease diagnosis in underdeveloped and developing countries. Another important suggestions from World Health Organization is that these sensors should comply the assured criteria. What is assured? Assured means affordable, sensitive, specific, user-friendly, rapid and robust, equipment-free, deliverable, and end users. And these criteria are very much useful for the sensors for their deployment in point-of-care applications or remote locations. Now let us look at the application potential of the biosensors. The application of biosensors is diverse in diverse area. For example, in disease detections, environmental monitoring, soil quality monitoring, food quality monitoring, toxin in defense interest. Now this is one of the very emerging area because biological warfare is to detect different kind of toxic or different kind of highly contagious microorganisms like Ebola, anthrax, even coronavirus in a area like airport, railway station, and all these things. Currently, sniffer dogs are used, but to detect those kind of biological material is very difficult for dog also. Water quality management, the detection of heavy metal toxic compounds in water is very important. Prosthetic device, if a person suppose does not have his hand, if he used the prosthetic hand, then these hands should be able to sense the material what he touched, and for that a chance of sensor is required. So there are very wide range of applications, and currently another area of biosensors is emerging, that is wearable biosensors. These wearable biosensors relies on the non-invasive fuels like saliva, transdermal fluid, sweat, tears, etc. One important advancement on this area is the Google's contact lens. Many of you might have heard. So Google contact lens is nothing but it is an enzyme-based biofuel cell-based biosensors. It is implanted on the surface of the contact lens and it produces power about 1 milliwatt per square centimeter at 0.5 volt. So this actually run with the glucose which is present in the lacrimal fluid. So more the glucose, more will be the power. And this power, this current can be, signal can be transferred to through different communication technology like RFID to your mobile phone and remote locations. And from there, one can identify how much glucose is there in the body. So this is one non-invasive technique. The other important area of wearable biosensor is that it is very useful for elderly persons. The elderly persons which are in the old age home or in your own home, to monitor the elderly persons from remote area, for example, from that office. Different kind of sensors can be put on the surface of the body, like one can measure the blood pressure, pulse, then ECG, and various kind of sensors, leg movement. Many things can be implanted on a cell surface, on the body surface. And this surface can be connected to your mobile phone through Internet of Things. The Internet of Things is a modern technology through which without any human intervention, the signal can be transmitted from that body to the remote locations. For example, if it is for clinical applications, then that can be transmitted to remote centralized laboratory where doctor or nurse sits. They can analyze the data and then give the feedback, assistance and therapeutic intervention, whatever it may be. This is one of the very emerging area. So particularly this research is very intensively going on in developed countries. But today I'll be talking about the potential area for developing biosensors in Assam. So obviously one of the important areas is the healthcare, and lifestyle disease is obviously one of the very important area. such as cancer, cardiovascular, diabetes, etc. These diseases are everywhere in all countries, whether it is developed countries or developing countries. But recently, cancer in northeastern India, it is a big news because northeast India has the highest incidence of cancer in the country as per ICMR report 2020. The northeast is the cancer capital of the country as per the Times of India February 5, 2020. And in most of those cancers, there are various kind of cancers are there, and most of those cancers are generally diagnosed by using biopsy technique. Another important lifestyle disease is the stroke. The alarming in Assam, a study conducted by NRC Hospital Guwahati during last 17 years has revealed that as many as 4,000 people are detected with heart disease, while 48 die of stroke every day in Assam. So apart from this lifestyle disease, the developing and underdeveloped countries has another disease burden. What is that? This is mainly infectious disease. This is a serious health problem caused by malaria, diarrhea, Japanese encephalitis, and various skin diseases. Particularly, the skin disease in Assam is well known because the Assam in the northeastern region, due to the high humid climate, the fungal growth is very predominant here. So due to that fungal growth, various kind of fungal as well as bacterial growth, this kind of skin diseases occurs. Another important infectious disease, which is primarily not in news, but it is creating a great problem. It is a very serious issue. That is diarrhea. It's primarily caused by virus and bacteria pathogen. Assam recorded the highest number of deaths, 439, due to diarrhea in 2018, as per the Central Bureau of Health Intelligence under the Ministry of Health and Welfare, Family Welfare. So another important issue is the toxic heavy metals in Assam, in different parts of Assam, whether it is the Brahmaputra Valley or in a Barak Valley or in a Karbi Anglong. Particularly the arsenic, mercury, lead, cadmium, chromium, iron in different parts of Assam are frequently in news. For example, cadmium in Dhemaji, Kamrup district of Assam and Shillong in Meghalaya. Arsenic, it is in Karimganj and Dhemaji, Jorhat, Golaghat, Lakhimpur, etc. So these serious health issues need timely and rapid screening facility for emergency therapeutic intervention, disease management, and prevention of this disease uh, on the right time. So the biosensors has another important area to develop in Assam, is agriculture and food. The quality testing of tea. As you know, the Assam is known for tea, but currently the tea testing, it is one of the important activity in tea industry for its business, is mainly done by human sniffing or different kind of electronic nose. So most of those techniques are not very precise, not very specific. So for that, some specific kind of sensor is required to understand the flavor of different teas, alcohol detection, formaldehyde in fish and other food items. This is already in the news. Adulteration in milk, such as detection of urea, methanol detection. I think bootleg alcohol, as you know well, in 2019, many people died in Golaghat in Assam. Toxins, various bacterial and fungal toxins in our foods, pathogen in foods and water, etc., etc., etc. It is already we know it is very serious issue in Assam. Now, one of the important issues, the methanol poison. It is called bootleg alcohol. There has been frequent cases and mortality in Assam, particularly within the tea garden laborers and less educated and economically poor fraction of the society. For example, in 23rd February 2019, in Jorhat, more than 150 people have died and 200 more were hospitalized. So this is a very serious issue. Now, if we see all these issues starting from lifestyle disease, infectious disease, and agriculture and food, if you see the rapid detection tests, there are very few rapid detection tests are available in our local market. Although many of the rapid detection tests are available in the global market, but they are limited in our local market. And the other issue is that among the sensors or rapid detection tests which is available in the local market, many of them are not very reliable or they are very highly expensive. For example, alcohol detection. Alcohol detection, we are mostly focused on breath analyzer, which are mainly used for law and order situation by the traffic police. There is no suitable alcohol sensors for detection of alcohol in liquid sample. So currently there is one urine alcohol test device is there, but it is highly expensive, about 2,875 per box. So there is a great demand of developing many of these biosensors or rapid detection tests in the context of Assam. So R&D work at Biosensor and Biofuel Cell at my lab at IIT Guwahati are focusing some of the important targets. One of the malaria detection, then early detection of myocardial infection, which is normally called heart attack, alcohol detection, methanol detection, formaldehyde detection, and the rotavirus detection that is mainly cause the diarrhea. So main objective of our research work at IIT Guwahati in my lab is to develop the low-cost, reliable, disposable, rapid detection kit or biosensors. Now, one of our focus area of developing these biosensors is the low-cost detection of the device and disposable detection device. And for that, we find chromatographic paper as a sensor platform or as a detection platform is very suitable. Because paper, as such, it is available and it is inexpensive. It has passive transport of fluid because in paper, liquid flows through capillary suction. There is no active transport is required, so you need not have to apply any external force or pump to flow the liquid on the surface of the paper. Papers are thin, lightweight, easy to stack, store, and transport, compatible with biological samples, easy uh, fabrications, good medium of colorimetric test, can be disposed by incineration. So this disposal of clinical sensors or medical waste is very important thing. Currently, there is no suitable disposal technology. But if we use paper kind of platform, then those used medical sensors can be disposed by incineration easily to kill the germs and remove all these contaminations. Compatible with a host of existing printing technology. So paper is compatible. On the surface of the paper, you can print the reagents, you can print the electrodes. So this printing can be done by various existing printing technology. Now, question is that if you use paper, then if you drop a sample of RDR, then sample will spread across all the sides of the paper. So in that way, sample will be diffused. So it is very important to design a template, to design a pattern on the paper, so that where you drop the samples, it will be not diffused out to the surrounding. So for that, a hydrophilic zone is need to be created, and surrounding this hydrophilic zone, a hydrophobic zone is created. For example, for detection of glucose, this kind of pattern can be used where one is sample application zone, second one is the test zone, absorption zone, third one is the test zone. So in the test zone, you are incorporating all your reagents, for example, glucose oxidase and horsedase peroxidase. And when you load this sample will move only through the hydrophilic channel. And then ultimately it will react to the reagent which is present in the test zone. And as a result of that, if there is a glucose, that will react with all those reagents and that color, one compound is used, that compound will be converted to different colors. So that based on the color or its pixel intensity, the presence of glucose or its quantity can be detected. Now, in our laboratory, we have used one technique to develop this pattern that is called flash technique, fast lithography activation of sheet. By using this technique, we develop the pattern on the surface of the paper to create that hydrophilic and hydrophobic zone. And from there, we can develop the pattern. Now, one of the important issues is that when you use paper, in the rear side of the paper, the sample leaks out. So to prevent that technique, we have developed one technology so that this sample, when you load on the surface of that strip, it will not leak out from the rear side of that paper. Additionally, we have also developed one technology that sample the real test zone, the test zone where reagent will be there, that reagent test zone can be separately prepared, and at the time of this test can be put on the surface of the paper. So in that way, you can keep a large number of test zones separately in a low temperature if it is required. Then at the time of use it can be used. So it will help in stake and transportation of the paper-based uh, strips. So basically, this kind of paper-based strips has detection time about 20 seconds. Sample volume only 7 microliter sample is required. It is a leak proof. It has prefabricated test zone as I have already discussed. So it prevents sample and reagent loss, facilitate transport, storage, easy loading of the reagent, etc., etc. So this already we have filed 2 patents on it. So one of the important target, as I have already mentioned, is the paper-based alcohol biosensor. Now, question is that why alcohol biosensors? And I have also mentioned that we are not targeting that alcohol biosensors for traffic or law and order applications. We know that in the northeastern region, in different parts, the tradition of drinking homemade alcohol is common among the tribes and tree garden community in the northeastern state. Even in Arunachal itself, more than 16 varieties of man-made alcohol preparations are here, but these local varieties are not able to penetrate the market due to lack of standardized method of large-scale preparations and absence of simple monitoring device to prove the quality. If a large-scale production cannot be done, and if when fermentation is going on at that time, whether the alcohol which is produced is the right flavored alcohol or not, to understand that thing, you need a simple testing device. Time to time, it is need to be tested. So those kind of technology are lacking. As a result of that, even though many of the preparations has high potential for market, is a high potential to attract the customers, but it could not penetrate the market so far. So we want to develop a sensor for detection of alcohol for fermentation industries. And as a first step, what we are doing, we are using a bacteria as a recognition, biorecognition element on a paper surface. Why we have selected cyanobacteria? Because cyanobacteria, it does not require any other carbon source other than carbon dioxide for its growth. It can grow in other carbon sources also, but it can go simply by using the carbon dioxide from the atmosphere in presence of light and small amount of mineral. So its growth is self-sustained without much human intervention. So that means it can grow on a solid surface also. So it is tolerant to high salt and organic contaminants. Generally, cyanobacteria are known to tolerant to the high salt and organic contaminants. So if your sample contains some other toxic compounds, that will not much affect the sensor performance. So cyanobacteria, by using cyanobacteria, we have developed a biofuel cell-based paper strip sensor by developing one pattern, the hydrophilic zone and hydrophobic zone. Inside it is the anode and outside it is the cathode. In the anode, we immobilize these bacteria by using some nanocomposite matrix. And by using this biofuel cell, we could detect the alcohol. More the alcohol in the sample, more the spike the potential. And the spiking of the potential can be plotted against the potential, against the concentration of the alcohol, and from there, linear dynamic range can be derived. So response time is within 10 seconds, and linear lowest detection limit is the 0.02%. So the work is going on to transform it to a technological device. Now another two products which are developed in my laboratory, and recently these two products has been dedicated to the nation by our Honorable HRD Minister in the last 14 February 2020 at IIT Guwahati. One is for methanol sensing. As I have already mentioned, that methanol sensor is very important, not only of course in the Assam and northeastern region, but in different parts of our country. There are many cases, year-wise, there are many cases of bootleg alcohol. Many people die. Recently, there are some cases in Punjab, and before that, some cases in UP. And in last year, it was many cases in Assam, as I have already mentioned. So we have developed a simple methanol detection kit, and the detection limit is far less than what World Health Organization has recommended, 200 ppb. So it requires time around 120 seconds, less than 120 seconds. Sample volume is only 1 microliter is enough. Cost is less than 5 Indian rupees. So that we have filed a patent. And second product, we have developed a formaldehyde sensing kit. So that also it is far less than World Health recommended by World Health Organization, lowest detection limit 200 ppb. But it requires time far lesser. It requires only 30 seconds. Sample volume is only 1 microliter and cost is less than 5 Indian rupees. So for that also we have developed the patent applications. So these products are already ready for commercial applications. And another product we have developed, that is the paper-based kit for detection of malaria. Now for this paper-based kit, we have developed two aptamers. Aptamers are here, what we are developing is small nucleic acid based aptamers. It is called DNA aptamers. One aptamer can specifically recognize lactate dehydrogenase as a biomarker for pan-malaria detection. This is lactate dehydrogenase is present across all malarial parasites. So this aptamer can specifically detect that lactate dehydrogenase. And the other aptamer we have developed that can specifically detect Plasmodium falciparum glutamate dehydrogenase. So if disease is caused by Plasmodium falciparum or not, can be easily detected by using these aptamers. So by using these aptamers, we have developed a paper-based strip. We use their magnetic nanoparticles. On the surface of that magnetic nanoparticles, we immobilize these two aptamers or one aptamer at a time. And when this magnetic nanoparticle is dipped in the blood serum sample, it will capture this biorecognition element, Plasmodium lactate dehydrogenase or Plasmodium falciparum glutamate dehydrogenase, and then it can separate out, and then these captured biomarkers are then allowed to react with a cocktail dye. And if there is these two enzymes are there, basically these two biomarkers are two different enzymes, then the catalytic reaction will be there, and that will convert the blue color of the dye into a pink color. The intensity of the pink color is an indication of the level of infections. So this whole reactions can be carried out in a simple small syringe itself. So now this kit which we have developed is now going to, we are using for clinical validation. So this work is already in different newses, even in the Hindu, in Nature India, and the work has already been published, and the work has already been patent. We have also filed patent application for both these biomarkers, aptamers which we have developed for both the biomarkers, and the reactions which we have developed for the kit. Another kit we have developed, that is the proof of concept for malaria detection, that we termed it as OptiSense. It is a smartphone-based multi-channel static localized surface plasmon resonance system for detection or diagnosis using optical fiber as support system. So we could couple optical fibers in the beneath of the smartphone, and then on the optical fiber, we would immobilize these two aptamers already, which I have discussed in the previous slide. And these two aptamers, after immobilization, if in a sample, these biomarkers are there, these will be captured here. As a result of this, the optical signal will be changed, and that optical signal can be read in the form of pixel intensity. And the intensity, based on the pixel intensity, the severity of the disease could be understood. So this work has already been patent application filed, and then subsequently we have published the work. So at the end, I would like to present you a book recently written by us, edited by us. This is Advanced Material and Technique for Biosensors and Biofuel Cell Applications. So this book is, I am expecting that in the coming month, sometimes in the coming month, it will be in the market. Many companies are advertising this book. The cost is they are proposing around 155 pounds. So this book maybe personally it will be difficult to procure, but it can be purchased through laboratory. So I request you all to subscribe this book in your laboratory if possible, because this book will give you enough information about what is biosensors, how biosensors can be fabricated in practically, and what are the different kind of advanced materials and techniques are used for developing biosensors, an emerging area of biosensor research in the global science. With this, I would like to thank my all respected audience for listening my talk, and I would like to acknowledge my research groups and my collaborators, Professor Melram from Canada, Professor Estrella from University of Bath, UK, Professor Corton from Argentina, and Professor Stom for Scott State University of Russia. Thank you very much.